Good morning, everyone. This is Pratik Kulkarni, PhD candidate, final year, awaiting final defense from India at National Forensic Sciences University. I would like to first of all thank LabRoots team for this wonderful opportunity. Uh, Next, I will introduce myself and what I do and how my university functions, and then I will directly dive into the topic, which is Niosomes as a bilayer nanocarrier for combinative drug delivery. As we move on, I will just start with a brief pictures of a wonderful university. It is a National Forensic Sciences University, as I said. It is an autonomous university, one of its kind, and it's the first university is dedicated to forensic sciences in the whole wide world. And I am specifically in the Department of Research and Development, catering to the applications of drug delivery and nanotechnology. I have been guided by my supervisor, Associate Professor Dr. Deepak Rautani, who has helped me in my research career in developing these novel ideas for my research and garnering applications for different diseases. So let's start with the topic. So my topic is Niosomes as a bilayer nanocarrier for combinative drug delivery. So today I'm going to talk about a few things about what actually are Niosomes. Some of you might be already aware about it. Uh, Different kinds of nanocarrier drug delivery and why specifically combinative drug delivery has its significance in the current scenario of therapies and treatments. Also, I will shed some light upon the recent examples on nanocarrier-based combinative therapy. And finally, I will be discussing my own research in this specific topic and how I achieved those goals in two different disease therapeutic areas, that is tuberculosis and breast cancer. And finally, we will discuss about the summary and future directions as to where this research will lead to the future students and what might be expected out of this in the longer term. So, as I said, Niosomes might be aware to some of you, but let me tell you and define it again for you that Niosomes are nothing but non-ionic liposomes, which are self-assembled amphiphilic nanostructures formed when a non-ionic surfactant and polar lipids such as cholesterol are mixed together in a specific ratio. As it is a bilayer structure, it is obvious that we can use it for entrapping lipophilic as well as hydrophilic drugs in a single vesicle. This has been also previously achieved with liposomes. However, there are some issues with the liposomes which we will figure out here and give the advantages of niosomes specifically over those niosomes just to advance or look forward for their better industrial applications and patient care. As I said, use of liposomes have been there for various time, including cancer has been reported. However, some issues like low physical and chemical stability, higher micellar solubilization, and higher cost of phospholipids, which is a bigger issue, and also stability and difficulty in surfactant derivatization are some of the main issues which we encounter with preparing liposomes, and also lack of method optimization for its preparation is also a big concern for liposomes. And compared to that, niosomes have showed a better control over these issues and has really shown some good potential for future therapies itself, which I think which may uh, replace these liposomes and can be used for a better application uh, in the world. These are some schematic diagrams of what actually a bilayer looks like. The outer portion, as you can see, is a lipophilic portion where It can be used for entrapping lipophilic or water insoluble drugs, and the internal aqueous core, the bigger circle inside, can be used for entrapping water soluble or hydrophilic drugs. The method of preparation varies accordingly for both liposomes and niosomes, and I will also discuss how we optimize the specific method and achieve the formation of niosomes in a much better, shorter time and better control over the other characteristics of the vesicle. So let's discuss the main thing here, that why combinative drug delivery is important here and its current significance in the scenario right now. So treatment of many diseases, as we know, require multiple doses of one or more drugs in a combination, which you might be aware of, you taking too many tablets at a time or during the day. So which ultimately in the long term causes increased side effects, interactions, dosage of drugs also increases time to time. There is a failure due to that. The failure in the complying of the patients is there, and also there is a threat of recurring of the same disease or a secondary infection or a disease. And to counter that, combination therapy is used to tackle these above issues, which directly reduces, first of all, the duration of the therapy, also the side effects, and ultimately the drug efficacy, bioavailability, and drug distribution to the target is significantly increased, which shows a better profile of the drug and safety also. And for that, nanocarriers like liposomes, niosomes, and many other lipid nanocarriers like solid lipid nanoparticles, micelles have been used extensively for application in the treatment of various however, their industrial applicability is very low due to the stability and cost constraints, and I mean scaling up to the industrial level, it fails a lot. So this is the main issue with lots of nanocarriers and nanomedicine in general. Now, as I said, there have been extensively studied in the past, and there are actually several promising reports in the recent times which have showcased the true potential of such nanocarrier systems, specifically for combinative drug delivery, which could change the paradigm of this disease therapies. And the basic reason for utilizing these therapies is to counter these issues and provide a sustained relief for a longer time, which ultimately, as I said, decreases the therapy duration and decreases the side effects and significantly increases the drug efficacy and action. And also, it tackles the issue of patient suitability and compliance. And also, as compared to liposomes and other lipid carriers, the niosomes, uh, as per my research, have been providing a better stability, which increases its industrial prospects as well. There are some few examples of combinative drug delivery achieved by using nanocarriers or specifically lipidic nanocarriers. In a recent study by Varsha Sharma and the team in 2015 introduced the concept of self-degrading niosomes, which entrap both hydrophilic and hydrophobic drugs in a single niosomal system or in a single vesicle for anti-cancer applications. And the results showed that the drug efficacy was significantly improved when compared with the free drug combination, which shows the true potential of this study. In a similar study carried out by Samed et al. And team in 2018, dual drug-loaded niosomes comprising of glipizide and metformin were formulated as an anti-diabetic formulation, and again the results showed a better drug profile over free drug combination, and also showed a sustained drug release of up to 14 hours and higher drug entrapment efficiency, which correlates with the decreased duration of therapy and increased drug effect or action. And also, there have been some applications in the areas of oncology like breast cancer, infectious diseases like tuberculosis, and also uh, some have some have been reported that it also possesses some antioxidant applications. So from this onward, I will start with my own research as how I achieved using this specific nanocarrier called Niosomes and uh, proved the conceptual validity of the combinative drug delivery in the areas of breast cancer and tuberculosis specifically. So for cancer, I selected specifically breast cancer, where a very common combination of drugs is usually prescribed by doctors, which includes the drugs that are tamoxifen and doxorubicin, cyclophosphamide, etc. So just for our research, we selected one uh, hydrophobic drug and hydrophilic drug, which acted as tamoxifen as hydrophobic and doxorubicin as hydrophilic drug. And for the another project that is tuberculosis and niosomes, we selected the drugs ethionamide and D-cycloserine, which are mostly used as a combination as per the recent WHO 2016 update, which states that using a combination of drugs results in a better treatment strategy over the long term. So this was the main reason behind selecting those drugs, and ethionamide and D-cycloserine were those specific drugs which provided lots of discrepancies in their antimicrobial efficacies, which propelled us to select these specific drugs and go forward with the research. So, to develop the plan of this work, which was the combination of drugs was uh, had to be done in the first time, and due because the first step of any formulation is preliminary studies, and after some uh, success in the preliminary studies, optimization studies and of the formulation has to be done using experimental designs and other techniques, which is followed by the characterization of the drugs using different analytical and advanced ultrastructural characterization techniques, and also as the drugs and the treatment therapy was for anti-cancer, breast cancer and tuberculosis. For cancer, we perform an in vitro MTD cytotoxicity assay, followed by the nanocarrier uptake study, which showed the potential of niosomes internalization in the cells and showing a better drug effect in comparison to the free drug combination. And for in the tuberculosis niosomes, we performed an in vitro minimum inhibitory concentration study. And for both the studies, we also performed the combination index, which uh, proved the synergism of this work and which can be used by the researchers for further studies as well for different applications and different therapeutic areas. So this is just a schematic of how the neosomes were prepared, and specifically this method called ethanol evaporation method was extensively optimized for the to gain the neosomes or to prepare neosomes in the shortest amount of time possible. It took around 2 years to us in comparison to conventional bilayer vesicle preparation techniques like thin film hydration methods. We selected ethanol injection method, which took around 2 to 4 hours for the complete preparation and homogenization of our neosomes, compared to thin film hydration, which almost takes around a day to prepare those bilayer vesicles. So it's a very simple method in which we use uh, the mixture of cholesterol and the non-ionic surfactant, specifically SPAN20, in a 1:1 mixture, and then we subjected it to a high-speed homogenization at around 8 to 9,000 RPM. And as you can actually imagine that both this organic phase and aqueous phase has to be mixed together. So as you can see, the small injection size the picture you can see that contains the organic phase, which contains the hydrophobic drug, and as well as the mixture of lipid and non-ionic surfactant. And this was mixed in a drop-wise manner at around 8 to 9,000 RPM with high-speed homogenization, followed by removal of the solvent at 60 degrees Celsius and storing in a refrigerator at 4 to 8 degrees Celsius for complete optimization, which took around 2 to 4 hours, as I said, for the complete preparation of niosomes, which were then further carried out for the stability study. And compared to this, this was the shortest amount of time we got for preparing niosomes compared to other procedures like thin film hydration and sonication methods. So the last initial successes in the preparation of niosomes and the stability of around 1 to 2 months. In the initial stage, we carried out the optimization of the formulation, which is one of the most important steps in the formulation design. And we used box Benken experimental design using design expert software, and in which the variables, independent variables and dependent variables has to be selected as to study which variables are affecting the size and entrapment efficiency of the niosomes, which were the dependent variables. So, for the ratio of cholesterol and SPAN20, ratio of drug, and the speed of homogenization were selected as independent variables, and the whole procedure was run for 15 formulations. We had to perform 15 different, we had to prepare different batches for the whole thing. And after that, the most suitable, the lowest particle size niosomes and highest entrapment efficiency for both the drugs were selected for further study, which will be just shown in the next slide. As you can see, the table here for both dual anti-cancer and dual anti-tuberculosis niosomes, there was a very high significance that we observed after the regression analysis for both the uh, projects. As you can see, the Y1 in the response section of the table is particle size of the niosomes, and the Y2 and Y3 are percentage entrapment efficiency of both the drugs, that is tamoxifen, doxorubicin for anti-cancer drugs, and ethionamide and D-cycloserine for anti-tuberculosis drugs. The ratio, the optimized ratio was selected to be 1:1 for cholesterol and SPAN20. The drug was 0.25 in comparison to 1:1 ratio, and the study speed was selected to be 8,500 RPM. Similarly, in the case of anti-tuberculosis niosomes, the drug ratio was even higher, that we were able to entrap even a higher amount of drug, which is corresponded to 0.375 mg. So that is to 1:1 ratio of cholesterol and SPAN20. And the results that were obtained were less than 200 nanometers for both the projects. In the case of dual anti-cancer niosomes, the optimized formulation had a vesicle size of 183 nanometers with an entrapment efficiency of more than 70% for both the drugs, as you can see in the table. And also in the case of dual anti-tuberculosis niosomes, we got even a better result, which showed that the niosomes had a particle size of 137.41 nanometers and a percentage entrapment efficiency of 96% in the case of ethionamide and 77% in the case of D-cycloserine. The next step was to characterize the niosomes after successful optimization of the formulation. The first and foremost were observing the particle size and beta potential of the niosomes, which were followed by drug excipient compatibility of the formulation, entrapment efficiency, and in vitro drug release profile of the drugs from the niosomes. Also, surface morphology of the niosomes were observed using microscopy techniques like AFM and transmission electron microscopy, and to assess the stability and safety of this formulation for IV purposes. Also, in vitro hemolysis assay was used, were performed to determine the in vivo biocompatibility. And in the end, we also performed osmotic shock study uh, to obtain biocompatibility in different media, and also as a in the form of different routes of formulation. I'll start first of all with both the results for both the uh, neosomes. This was the parameters evaluated. As I already discussed, that the dual anti-cancer neosome showed a particle size of less than 200 nanometers for 183.6 nanometer and a zeta potential of minus 26 MV with a PDI of 0.09, which is considered to be a very good PDI, which corresponds directly to the stability and homogeneous dispersion of the formulation. The drug release was for both the neosomes of therapeutic areas were up to 3 days, and robust stability of 6 months under refrigeration at 4 to 8 degrees Celsius was observed for both the neosomes. In terms of drug excipient compatibility using FTIR and DFT studies, there were no specific interactions found between the drugs and excipients, which shows the compatibility of the drugs with the excipients used, which were just lipids and the non-ionic surfactants. Uh, and also, the serum stability study showed no change in the absorbance of the solution, which corresponds to no turbidity and hence a good in vivo biocompatibility for IV usage. In terms of osmotic study, there was no change in the vesicle diameter at all in the saline and isotonic mediums. However, as expectedly in hypotonic and hypertonic mediums, the vesicle diameter was subsequently changed, which was expected. And in the cytotoxicity study, which was performed for dual anti-cancer neosomes, there was a 16-fold improvement observed over the pre-drug combination, which showed a significant synergistic effect and a greater nanocarrier uptake, which shows the potential of this dual drug-loaded neosomes for a better combinative therapeutic action. And for dual tuberculosis neosomes, 50% improvement in the antimicrobial effect was observed in this case too, in comparison to the plain drug combination, along with a good synergistic effect. Let's quickly go through some uh, microscopic images of the vesicles. As you can see, the AFM, the same images of blank neosomes, which shows a clear spherical structure and is in good correlation with the DLF spectrum as well. These are the same images of a dual anti-cancer neosomes, and you can clearly see uh, bilayer surface. I mean, bilayer which can be clearly visible in the neosome structure, which shows or confirms the entrapment of the drug and homogeneous formation of the neosome. These are the images, same images of the dual anti-tuberculosis neosome. Again, it also shows a good formation of bilayer around the vesicles, which correlates with a better stability and good drug entrapment. These are the images for FTR characterization for both dual anti-cancer neosomes and dual anti-tuberculosis neosomes. Compared to cholesterol and SPAN20 in the spectra, there was no peak observed for the drug in the spectra of neosomes, which shows complete entrapment of the drug inside the vesicles and no significant interaction of the drug with any excipient, which shows a good stability and compatibility. A specific observation can be seen in the broadening of the OH peak, as you can see. I have marked that thing here. This extension of the OH group can be seen due to the addition of water molecules while preparing the niosomes. Only the peaks of cholesterol and SPAN20 were visible in both the blank niosome spectra as well as the drug-loaded niosome spectra, which again correlates or confirms the drug entrapment and shows a good stability of the niosome. Next, I will show you the DSC thermograms and this characterization. And a very specific observation was found in this as well, that as you can see, specific exothermic peak at around 236 degrees can be clearly seen for all the niosome samples, which shows conversion of the drug into amorphous form, and this also correlates with the uh, good amount of stability and no interaction of the drug with the excipient, as there was no shift of any peak. Instead, there was no peak at all of cholesterol at SPAN20 visible, and which means after some extensive literature review, uh, this means that it has converted into an amorphous form and indicates a good stability and compatibility of the drugs with the excipient. These are the drug release spectra of both the studies. The first two graphs can be seen for the tamoxifen and doxorubicin loaded niosomes, and these were carried out at different pHs, at pH 7.4 to simulate the normal conditions, and at pH 5.4 to simulate the cancerous cell conditions. And as imagined, expected, the drug release was a bit faster at 5.4 due to higher pH, lower pH. However, the drug release was sustained. Uh, yes, due to sustained drug release, which was up to 3 days in both the cases. Similarly, in anti-tuberculosis niosome in the final graph, it was carried out at normal pH, and again the drug release was up to 3 days. For ethionamide, it was for 72 hours, and for d-cycloserine, it was up to 2 days. At the bottom, you can see the IC50 evaluation in the terms of microgram per ml. And as you can see, I have noted down the IC50 values of the anti-cancer niosomes, and you can clearly see the improvement of the IC50, which correlates with the anti-cytotoxic effect, and which correlates with the cytotoxic effect of the formulation. And the tamoxifen, the doxorubicin loaded niosomes showed an almost 15-fold improvement in the niosomes. The IC50 value, which shows very high cytotoxicity and a good effect. Followed, these are some microscopic images of the stain, of the MCT assay. It is quite well understood that with increasing concentration, the cytotoxicity was increased, which is clearly visible here. The upper right graph shows the three drug combination spectra, and the lower right shows spectra for tamoxifen plus doxorubicin loaded niosomes. This was followed by the nanocarrier uptake studies. Again, as expected, the nanocarrier, which is drug-loaded niosome, showed a better drug uptake in comparison to the blank-loaded niosomes, which shows or confirms that why such a higher cytotoxicity effect was observed. The main thing to observe here is that or the main inference from this nanocarrier uptake study can be taken that due to the drug loading or the entrapment of the drug inside the vesicles, the density of those vesicles are increased, which shows higher drug effect or efficacy or cytotoxicity, as you may say, and it clearly shows from the diagram that increasing uh, with increasing concentration, the intensity or the color intensity of the drug is increased and uptake is increased. The lower image shows the different areas of the cell. The blue image shows the uptake in the nucleus section. The green and red shows in the cytoplasmic section, and this is the merged image which shows the increased intensity of the dual drug loaded niosomes, which correlates and corresponds to a better cytotoxic effect. And the B spectra shows the flow cytometry analysis in the quantitative way, which proved that due to increased drug loading, there has been an increased drug uptake or the nanocarrier uptake in the cells, which indicates a better cytotoxic effect to the cell. This is just a summary of the anti-cancer niosomes, where it shows the before the treatment of cells before treatment and after treatment with the niosomes. This is before the label of this is the FIDC labeled spectra of blank niosomes compared to the FIDC label of dual drug loaded niosomes, and you can clearly see the difference in the intensity of using both qualitative and quantitative results. This is the stability of the formulation starting at day 1 and at day month 6 months. And finally, for anti-tuberculosis niosomes, as I said, an MIC study was done to evaluate the antimicrobial effect of the dual drug loaded niosomes, and expectedly here too, we observed a better antimicrobial effect for the dual drug loaded niosomes compared to the free drug combination. And as you can see, the free drug combination showed a 62.52 microgram per ml MIC, which was 50% higher than the MIC of ethionamide plus cycloserine loaded niosomes at the number 2, which showed 50% lower MIC, which indicates a better effect and a lower amount of drug needed to be used for the treatment. We also, for a good amount of comparison, we also performed uh, the same analysis for all the studies in terms of single drug loaded niosomes as well, just to get a better comparison. And finally, combination index values were uh, studied or evaluated to, calculate the synergistic effect of the formulation. As you can see, the combination index value of the niosomes is comparatively lower to that of free drug combination, which indicates better synergistic effect. And finally, the osmotic shock study shows uh, the visual inspection of the vesicles in different media. As you can see, the in hypertonic medium in the first image, upper and lower, you can see the shrinking of the vesicles in the hypertonic medium, and subsequently, as we move forward in the slide, that is CBS and isotonic media, the vesicle retains its size, which indicates its compatibility in different media and for different routes of administration as well. And again, with the hypotonic, you can see the bulge structure of the vesicles, which eventually got burst due to the hypotonic medium. The highlights, as we come to the end, we will discuss the highlights and summary and some future directions of this project and where it might be. The main highlights of the project were that both hydrophilic and hydrophobic drugs were entrapped in a single neosome system for applications in the treatment of breast cancer and tuberculosis. And in both the studies, the particle size of the niosomes was less than 200 nanometer, and the higher and the entrapment efficiency for both the projects were more than 70% for all the drugs used. Also, it showed a significant synergistic effect in terms of their free drug counterparts, and also they showed a good amount of sustained release for 2 to 4 days with a 6 month stability under refrigeration, which is quite a good thing in a liquid form. With coming to the end, we will discuss the summary and future directions that niosomes can be potentially used now that as a self-assembly based nanocarrier for entrapping multiple drugs within its bilayer and for using it as a therapeutic drug delivery option for many various applications. This can be used for combinative therapy as well as different routes of drug delivery is envisioned by your preparing this specific kind of niosomes using the ethanol injection method or any other method which the researchers might use in the future. Also, in lieu of the traditional therapies, it is envisioned that this combinative therapy using niosomes or any other lipidic carriers is a way for a better and smarter treatment option in the coming time. The future and some limitations of the study is that uh, use of some smart molecules for targeting specific areas in the body may provide a balanced option for the treatment. Also, using uh, better in vivo model and using these niosomes for performing better in vivo studies. And just to confirm their validity in the in vivo system can be done and must be done for a better therapeutic outcome and better scalability in terms of industrial application. Also, the intracellular state of these niosomes must be done in the future, and the underlying mechanism of it must be studied as to why they remain stable in the system and how they specifically act on the cells. A small amount of that we have seen was done in the drug uptake studies where the neosome uptake was carried out or was studied using both fluorescence microscopy and flow cytometry studies showing that with drug entrapment, the density of the vesicles gets increased and ultimately increases their uptake in the cellular areas, showing a better drug effect and action. In the end, this neosome-based carrier can be used for further applications in different areas as well. There are also some applications coming out in food technology and even in tissue engineering and the development of bioscience. These are some references for my presentation. Both these studies have been successfully published in Elsevier journals. One has been published in Journal of Pharmaceutical Sciences, which was for the anti-cancer newsrooms and was selected in the virtual issue titled Most Original and Most Significant Scientific Contribution. And tuberculosis work has been published in Colloid Surfaces A journal. There are also some other publications in work, and I also just published recently, 2 days back, of the same concept we have used for the newsrooms for application in the Alzheimer disease treatment, which we will share with the team after I get the official link of the publication. These are some other publications of mine, and this brings me to the end of my talk. I would specifically like to thank the LabRoots team for giving me this wonderful opportunity and a platform to uh, present my research and share some ideas for the fellow researchers and audience. So thank you very much for listening to me, and I would love some questions about this research, and I would love to contribute in any way. If you want to talk with me or you want to contact me, you can directly uh, connect with me on LinkedIn, I can share you that. So thank you very much.